Welcome to Chapter 14. Chapter 14 talks about blood, so we'll be discussing blood cells and their functions. First, a little introduction about the blood system. The blood is the only connective tissue that is made out of a liquid matrix. And within that liquid matrix, you have these cells, your red blood cells, platelets, and white blood cells that are circulating within that matrix. It is important for transportation of vital substances like vitamins and minerals and hormones. It maintains the stability of the interstitial fluid. Blood is responsible for distributing heat. Okay, and know that your major heat producer would be cardiac muscle activity. And through the pumping, that the blood will take that heat that was produced by the heart and distribute it to the rest of the body. The amount of blood varies from one person to another. So on average, an adult would have about 5 liters of blood. But it differs according to body size. It differs according to electrolyte concentration. So if somebody has more sodium or more salt intake, that would obviously lead to a higher blood volume. It also is related to gender. So generally speaking, females have about 4 to 5 liters, while males would have a little bit more, 5 to 6 liters of blood. And that is due to the loss of blood on a monthly basis in females due to menstruation. Now for the blood cells are made in the red bone marrow. Okay, and they're known as formed elements. So we're going to be talking about these formed elements. Know that when we say formed elements, we mean your red blood cells, white blood cells, and platelets. And we call platelets cell fragments because they are really not a cell in the meaning of having a cytoplasm and a nucleus. They are rather just cytoplasmic fragments from a bigger cell, and we will learn about that in this video. So you can see here, we're taking a little blood sample, and you can see what it looks like. We'll put it in a centrifuge. A centrifuge is a machine that spins around really fast, kind of like a dryer, and then we'll take the tube out after the centrifugation occurs, and you can see what the blood looks like. When you do that, you're separating the formed elements or the cells. They will go down by gravity. So you can see here your red blood cells. You can see on top the plasma, and then in the middle there's a little buffy coat that has the white blood cells and platelets. So this is what it looks like when you look at a centrifuged blood sample. But if you look at a peripheral blood sample where you take a little drop of blood and you spread it on a slide, on a lab slide, this is what you see. So you'll see, you can get a count of the red blood cells, a count of your white blood cells, and platelets. So looking at the centrifuge blood sample, you'll find that 55% of blood is made out of plasma, while 45% of blood is made out of red cells, and then the buffy coat is usually less than 1%. Okay, and you can see here that the red cells here, we're giving it in a percentage form, not in a count form. So we're not counting how many RBCs we have. It's a percentage of the RBCs in relationship to the total volume of blood. And that percentage of RBCs is known as the hematocrit or a packed cell volume. So again, hematocrit or your packed cell volume is a percentage and not a number of RBCs. So what is the composition of blood? Well, we already know that it's made out of 55% plasma and 45% formed elements, mainly RBCs. So let's look at the plasma first. That plasma is made out of more than 90% of water. And then within that water, we have things that are swimming there. So we have electrolytes, so different salts. We've got proteins. Most of these proteins, the majority of them are made by the liver. So we've got albumins, globulins, and fibrinogen. There are waste products like carbon dioxide in order to get rid of those waste products. There are nutrients, vitamins, and hormones. And there's also gases. So we know about oxygen and carbon dioxide, but there's also nitrogen. Okay, so all of these substances here are dissolved in plasma. For your formed elements, the majority of the formed elements, about 95% of them, are red blood cells, followed by platelets, and then white blood cells. So although we have 5 different kinds of white blood cells, they actually make the very minority of your formed elements. So let's introduce the type of the white blood cells. We'll go into the details of them later on. We have granulocytes. Granulocytes means that if you look under a microscope, you will find granules, and those are neutrophils, eosinophils, and basophils, and the names are based off of the color of those granules. So if the color is neutral, they're known as neutrophils. If the granules are red in color, they're known as eosinophils. If the granules are blue, they are called basophils. And then we have 2 other different kinds of white blood cells that do not have granules, and those are known as agranulocytes. They're monocytes and lymphocytes. Okay. In regards to these numbers, you want to know that the neutrophils are the majority of our white blood cells, whether we have an infection or not, and the minority would be your basophils. Now, since we're going into the field of healthcare, we have to be you want to be familiar with these universal precautions. Universal precautions are a set of safety measures that every healthcare provider would have to implement to make sure that they are not transmitting blood-borne infections in the workplace. So whether you have the pathogen, you don't want to transmit it to the patient or your coworkers, or it's the patient that has the pathogen and you want to protect yourself and your family and your coworkers. And universal means that you are going to assume that any patient that comes in is a source of infection. Okay, so you are going to put these standards into place whether the patient is known to have an infection or not. So regardless of the patient you're dealing with, you're going to be using gloves. You're going to implement all of these recommendations. And it's mainly used to prevent the spread of HIV, hepatitis B, and hepatitis C. Okay, but obviously it applies to other infections as well. And it's estimated that on average, about 5% of new cases of infectious diseases were transmitted due to unsafe injections. These specific recommendations, and you could read more on this if you're interested, would be the use of PPE or personal protective equipment like gloves and masks, and to use safe work practices such as hand washing. Now for the origin of blood cells. The formation of blood cells is a process known as hematopoiesis. So hemato comes from the word hematology, or anything that has to deal with blood. And poiesis means a synthesis, so your formation of blood cells. And they originate in the red bone marrow from stem cells known as the hematopoietic stem cells. Now, these hematopoietic stem cells are going to specialize, meaning that they're going to mature a little bit and grow up a little bit and make more specialized stem cells called lymphoid and myeloid stem cells. Lymphoid stem cells are going to give rise to lymphocytes only, while the myeloid stem cells will give rise to all of your other kinds of cells. So let's take a look at the process of hematopoiesis. So again, you start off with a hematopoietic stem cell, and then through different growth factors, that hematopoietic stem cell is going to be stimulated to differentiate into either a myeloid stem cell or a lymphoid stem cell. So a stem cell is a cell that basically has the ability to produce more than one kind of cell. So the hematopoietic stem cell has that ability to produce any kind of blood cell. But once it differentiates either into a myeloid or lymphoid, now they're kind of limited on what they can produce. So a lymphoid stem cell has the ability to produce either a T or a B lymphocyte. while the myeloid stem cell can produce either a red blood cell, platelets, eosinophils, basophils or neutrophils, and monocytes. So I kind of put that red line in there to so that anything on this side are produced by myeloid stem cells, and the two cells on this side are produced by the lymphoid stem cell. Now we want to go over the production of these individual cells. So we'll start off with the myeloid stem cells. Myeloid stem cells can go through a process known as erythropoiesis, which is the production of erythrocytes. Erythrocytes is a fancier word for red blood cell, and you have to be familiar with both of those terms. You will find that your myeloid stem cell can become a proerythroblast. Know that any cell that ends with the word blast means that it is a baby something. So it is erythroblast. That means that it's a baby erythrocyte. When it grows up and differentiates, it'll end up becoming a mature erythrocyte. Now, these steps right here are happening in the bone marrow, in the red bone marrow. This portion right here is happening in the bloodstream. So what appears in the bloodstream is a reticulocyte, which is an immature red blood cell, and then it has to mature into this erythrocyte. If you get a blood smear and you look at it under a microscope, you usually have less than 1% of reticulocytes, while the rest are going to be these mature RBCs. If it's higher than 1%, that means that this person is producing RBCs rapidly, so fast, and that is why these reticulocytes are appearing now in the peripheral blood smear. Okay. Moving on to your thrombocytes or platelets, so that myeloid stem cell again will become a megakaryoblast. So again, a blast is a cell that is going to become something else. So remember when we were talking about osteoblasts, it had to mature into an osteocyte. So a blastic cell is a cell that matures into whatever. So that megakaryoblast becomes a megakaryocyte. Mega means huge. So this is a huge cell. And remember we said that platelets are not really true cells. They are cytoplasmic fragments of a bigger cell. And that's what these platelets are. That megakaryocyte will pinch off some of its cytoplasm in order to make platelets or thrombocytes. And again, you want to be familiar with both of those names, thrombocytes and platelets. So your thrombocytes are cytoplasmic fragments of megakaryocytes. Now to make these white blood cells, the myeloid stem cells can make either eosinophils, basophils, or neutrophils, and these again are your granulocytes. Or it can also end up making a monocyte. Now these are the cells that appear in the circulating blood. So if you take a blood smear, these cells are going to be in that blood smear. But you can see here that monocytes have the ability to leave the blood vessels and go into the tissue. So you can see here activated in tissues in some cells, that monocyte is going to be will leave the blood vessel and become a macrophage. So a macrophage is pretty much a monocyte on steroids, where it is it's more active. It is highly phagocytic, means it'll eat up germs through phagocytosis. And you can find these macrophages within the interstitial tissue, whether it's in the liver or the intestines or wherever the infection is occurring. So a monocyte is the same as a macrophage, except monocytes are in the bloodstream, while macrophages are out of the bloodstream. Now moving on to the lymphoid stem cells, we end up making either T or B lymphocytes. B lymphocytes can also leave the bloodstream and change into plasma cells, and it's these plasma cells that will start producing antibodies. So plasma cells are antibody factories. And this ends this portion.